everyone, and welcome to another session in the general microbiology. And today we're going to be starting with two particular topics, one being the important scientists that have made some contribution in the field of microbiology. And another one that we're going to be talking about are the different types of microscopes that we use in microbiology, right? So, first we're going to be starting off with the scientist. So, let's begin. So, the first scientist that we are going to be talking about is your Anthony Philip van Leeuwenhoek, right? So, you can get sometimes direct pictures also of the scientist and you have to match them to their achievement, or you can just get their achievement and you have to just match it to the name of the scientist, right? So, Anthony Philips van Leeuwenhoek is also known as the father of, he is known as the father of microbiology. Right. So he's known as the father of microbiology. Why? Because if you have to do anything in microbiology, what do you require? You require a microscope. So what are you going to remember is that Philips is a company that makes a lot of electronic materials, and Philips also makes microscopes. Right. So he actually invented the microscope. And that is why he is known as the father of microbiology, because he invented the microscope. So under the microscope, when he saw the organisms, he called them what? He called them little animalcules. He called them little animalcules, which directly translated to little animals. Right? So that is the achievement of Anthony Philips van Leeuwenhoek, that he's known as the father of microbiology because he invented the microscope, and without the microscope we cannot see any kind of microbes. The world of microbiology would not exist, right? And how you can remember it, that Philips makes a lot of microscopes, and that is how the name Philips is there in his name as well. And he saw the organisms under microscopes and called them little animalcules, right? Okay, coming to the second scientist, that is the Louis Pasteur. So now Louis Pasteur is known as the father of modern microbiology. So father of microbiology was Anthony Philips van Leeuwenhoek, right? The father of modern microbiology is who? It's Louis Pasteur. Why? Because he actually made a whole institute, which is what? It is the Pasteur Institute in Paris. So he did a lot of modern research around that institute, right? So that is why he's known as the father of modern microbiology. So he actually made the Pasteur Institute, which is there where in Paris. So Pasteur Institute in Paris. Now, how will you remember the different achievements of Louis Pasteur? So from his name only, we're going to kind of devise his achievements. So just write L and P for Louis and Pasteur. So from L, what did he do? He gave us the concept of liquid media. So he told us that we can actually use liquid media for the isolation of bacteria, right? So that is the first L. Then from the name Pasteur, what did he do? He gave us the concept of pasteurization. Pasteurization is actually a sterilization method that we do for sterilizing milk to kill the microorganisms in milk. So from Pasteur, another thing that we will remember is the pasteurization. So pasteurization is kind of a sterilization method for milk, right? So that is the pasteurization. Then he gave us the concept of vaccine. So he actually invented 3 vaccines, and the mnemonic is going to be CAR. So what 3 vaccines? He invented the cholera vaccine, the anthrax, and the rabies, right? So one more thing that you can remember from this is that he used to, so from this car only you're going to remember that what kind of a car would he drive? He used to drive a G-Wagon car, right? And from G-Wagon you're going to remember what? That he gave us the concept of germ theory of disease. So germ theory of disease concept was given by whom? By Louis Pasteur, right? So those are all the achievements of Louis Pasteur that you can remember from his name itself, right? Okay. Coming to the next scientist is Joseph Lister. So he is known as what? He is known as the father of antiseptic surgery. So he's known as the father of antiseptic surgery. Why? Because he gave us the concept of using carbolic acid, which is kind of an acid to sterilize instruments and wounds. Right? So carbolic acid was kind of used as a antiseptic to clean the wounds and to sterilize the instruments back in his day. So he used carbolic acid to sterilize wounds and instruments. So that is why he's known as the father of antiseptic surgery, right? Coming to Paul Ehrlich. So Paul Ehrlich was another scientist in whose name only we're going to try and find his achievements, right? So first thing is that from the last CH, what are we? So there were two things. So CH, he's known as what? He's known as the father of chemotherapy, right? Chemo starts with the CH, so father of chemotherapy. So Paul Ehrlich is known as the father of chemotherapy because he gave us a drug called Salvarsan, which was used most commonly for the treatment of syphilis. So that is the first CH. Then what else did he do? Then he also gave us another concept which is known as the chain theory for antibody production. So he said that antibody production takes place in a form of a chain or a sequence. So chain theory of antibody production, right? So these are the two concepts with the word CH that is from his name. Now coming to Paul Ehrlich, we'll write it like that, and from Paul, from the A, we will remember two things, that he gave us the acid-fast nature of TB bacilli. Acid-fast nature, so we know that Mycobacterium tuberculosis is acid-fast in nature. That means it's able to resist sulfuric acid, right? So it is an acid-fast nature of the TB bacilli, which is seen due to the presence of the mycolic acid in the cell wall, right? So now acid-fast nature is seen by how? It is seen by staining techniques. So he also gave us a concept of staining techniques for tissues and blood cells, right? So that is your A from the Paul. Now from the Ehrlich. Now from the Ehrlich, what all did he do? He gave us three things. First, the Ehrlichia bacteria. So yes, there is an Ehrlichia bacteria which was kind of founded by the Ehrlich. Then there is an Ehrlich phenomena which tells us about the toxin-antitoxin reaction. That for every toxin, we can have an antitoxin. And then he also founded an institute which is the Paul Ehrlich Institute in Germany. Right, so these are all the achievements of Paul Ehrlich. Now coming to one another very important scientist, which is your Robert Koch. So Robert Koch, you're going to write his name as what? You're going to write it as Robert Koch, right? So again, from the S, what theory did he give us? He gave us the theory of solid media. So he gave us a solid media that he said that we could use agar as a solidifying agent, and then we could use solid medias for the culture or the isolation of the bacteria, right? Then from C, what did he give us? He gave us culture again. And what culture? He said that you could isolate organism in pure culture, right? Then from H, he gave us a method called as hanging drop, which helps us to do what? It helps us to check the motility of the organism. So hanging drop is kind of a method that is used to detect the if an organism is motile or not. So detect the motility of an organism. Then he gave he kind of discovered three bacteria, and you're going to remember it by the mnemonic CAT. So what are those three bacteria? C, the first one is your cholera, right? Then from A, it is your anthrax. And then from T, it is your tubercular bacilli or Mycobacterium tuberculosis, which is also known as what? It is also known as Koch's disease, right? So it's also known as Koch's disease or pulmonary Koch's. That's what we call it, okay? Then again, one thing from Koch. So taking things from his name only. One, we have already seen the Koch's disease. Other than that, he also gave us different concepts. So first, he gave us the Koch's phenomena. So in the Koch's phenomena, what did he say? He said that if you take a guinea pig who's been infected with tubercular bacilli and then you inject it with the tubercular bacilli, then you're going to see some level of a hypersensitivity reaction. So it's just, showing the hypersensitivity reaction, right? So that is your Koch's phenomena. Then he gave us Koch's postulates. So he gave us basically four criteria when it comes to the Koch's postulates. The first criteria that he gave us was that an organism should be constantly associated with disease. So suppose this particular rat, let's take it as a rat. So this rat has what? It has some lesion on his body, right? So he is saying that if it is caused by an organism, this lesion is the same lesion should be caused by this organism in any individual, in any animal, right? So it is saying that an organism should be constantly associated with disease. So suppose any organism produces an ulcer, then that is mean that that ulcer disease is something that should always be produced by that organism. So that is the first Koch's postulate that it gave us. Then he said that when I take this sample from this site, this lesion site, and I try to grow it in a culture medium, I should be able to isolate it in pure form, right? So I should be able to isolate in pure culture. That means only this organism should grow on the culture plate, which is associated with the disease, right? So it is only possible to isolate this organism in pure culture from the lesion. So isolated in pure culture. Now then he said that if I take this particular bacilli and then I inject it into another rat, right, into another rat. So what should happen? The rat should have the same disease. So the rat should have the same disease. So should produce same disease in another host, right? Then he's saying that now if I take, and now that it has produced this lesion, now if I take a sample from this lesion and again grow it in culture, it should grow in pure form. So should be able to isolate the same organism in pure culture. So again, that is the fourth postulate, that in pure culture, you should be able to isolate it, right? So these were the four Koch's postulates. So now there were certain exceptions, and these exceptions were mainly based on what? That if we did not have a media to isolate the organism, or if we did not have an animal model for that organism. So we have two particular organisms for which we don't have any media to grow them. We still don't have any artificial media to grow them. So we can't isolate them in pure culture in a media. So the postulate number 2 and 4 does not apply. Can't apply because we cannot isolate them in pure culture on an artificial media. And what are those two organisms? It is your Mycobacterium leprae. Right, Mycobacterium leprae, we don't have any media. And the second one is your Treponema pallidum, which causes what? Which causes your syphilis. So we are unable to kind of isolate them in artificial media. Now the second one is if we don't have any animal model. If we don't have any animal model, then your first and the third one doesn't apply, the Koch's postulate. So what animal model, so in what organism do we don't have an animal model? It is the Neisseria. So these are actually the exceptions, and you get a lot of questions on these, that what are the exceptions of the Koch's postulate. So these are the organisms that are the exceptions to the Koch's postulate. Then they said that no, it's not about the constant association of an organism with the disease. Other scientists kind of modified this Koch postulate. So they modified it by saying that it is not the organism, but some virulent gene that is associated with the disease. So they said that organism is not producing the disease, it is some gene in the organism that is producing the disease. So if that gene is present, so disease will be present. If that gene is absent, the disease should be absent. Right? So they are saying that this gene is associated more with the pathogenic strains than the non-pathogenic strain. So they did another kind of experiment in their self where they saw the gene virulence. So they said that if you mutate this gene, so if you mutate this gene, it is going to decrease the pathogenicity. That means it's going to decrease the disease production. Then they are saying that if you replace this kind of mutated gene with the wild type, that means the raw type that is present most commonly, then it will again produce the disease. Then it will become pathogenic. It will produce disease, right? Then they are saying that antibodies against the gene, against the disease should be present, and it should be the one that should protect the host. That means antibodies against the disease, against the organism, against whatever antigen is there, should protect the host, right? So, other than that, it said that it should be expressed at some point during the infection. So, just for an example, that initially, whenever there is infection that takes place, there is usually an incubation period where there is, not any expression of the gene, because at that time it is still getting used to. But once you get the clinical features is when it becomes pathogenic. So at some point, at this point, it is now causing infection. So at some point, it should kind of, be associated with the infection, right? And also an example of TB, where in a TB bacilli, initially in a tuberculosis disease, what can happen is that a person can have a latent infection, and during that latent infection, what is happening? That the person is clinically latent. That means clinically not producing any kind of disease. But when an immunocompromised stage is reached, what happens is that a person becomes clinically relevant. So clinical feature starts appearing whenever the immunity goes down and the TB bacilli becomes active. So now in this point where the immunosuppression has taken place and the TB bacilli is now active, this is the point where the infection or the gene is now expressing itself. So these were all the different modified postulates. So it just modified one and said that it is not the organism but the gene that is associated with the disease. Right, so that brings us to the end of the session on your scientist. Now we're going to be starting off with the microscopes. So in the microscopes, let's see, there are some properties of the microscopes. The first thing that I want you to know is that what is resolution? So resolution is actually the ability of a microscope to produce two separate images that are very closely placed. So example that if this was one thing seen under the microscope and this is another thing seen under the microscope, if I am able to tell that these two things are separate and they are very closely placed, that means the resolution is good of the microscope, right? So it is actually the ability to tell two closely related objects as separate, right? So different microscopes can have a different resolution or a different resolving power, right? So what is the resolving power or the resolution of the human eye? Human eye can actually see up to 12 micrometer or more. So 200 micrometer and more, organisms in size only a human eye can see. What about the LM? LM is the light microscope. So light microscope can see the organisms that are more than zero point 0.2 micrometer, right? More than 0.2 micrometer is the one that can be visible by the light microscope. Now coming to the electron microscope. So electron microscope can actually see structures that are as small as 0.5 nanometer. Now it has come to the nano. From micro, it has come to nano. Nano is even smaller than micro. So electron microscopes can see even smaller structures. That is why we are able to see viruses in electron microscope, because viruses are very, very small. They are less than zero point 0.5 nanometer, right? So we can see structures that are around zero point 0.5 nanometers. So in light microscope, it won't be visible. In human eye, it won't be visible. So that is why we want electron microscope. So these are the resolution or the resolving power of a microscope. Now coming to the contrast. So in contrast, what does it mean? That to increase the ability to view something. So that can be done by what? It can be done by staining. It can be done by staining. So stained specimens usually are better seen. So the contrast is increased. And what else we can do? We can either make some changes in the microscope and make a contrast enhance. So we can enhance the contrast by adding something to the microscope, right? And we will talk about what is it. Coming to the magnification. So magnification means if you have seen a microscope, you know that there is an ocular part. Okay, let me talk about this first. So there is an ocular lens in a microscope, and then there are the objective lenses, right? So these are the two lenses. So the ocular lens is actually the one which is known as an eyepiece, where you put your eyes and see, and the power for this is always 10x. Right, the power for this is always 10x. Whereas when it comes to the objective lens, what is the power? You can have three powers. You can have either 10x, 40x, and 100x. So these are the three powers that you can have at your objective lens. Now let's come to the magnification. So in magnification, what is happening? Like I said, your ocular lens has a power of 10x. Then your objective lens can have three powers, 10, 40, and 100x. So magnification is what? Magnification is your ocular power into your objective power. So if I have, focused some specimen at 40x, so what is going to be my magnification? So ocular lens is always going to be 10x, and if I have, put it on 40x magnification, then it is going to be on 40x objective lens. So my magnification is going to be 400x. So that is how you will calculate your magnification. Right? Okay. Coming to the different types of microscopes. The first thing I wanted to know is that there are two kinds of microscopes. One is the light microscope and one is the electron microscope. And why is the name like that, light and electron? Because the source is such. So the source of light microscope is going to be what? It is going to be the light. It's going to be the light. And what is the source of the electron microscope going to be? It is obviously going to be your electrons, right? So that is how the name are there. So light microscope source is the light, and electron microscope's source is going to be the electrons. So now light can be, given from two places. So either it can be a natural light, like from the sunlight, right? Or it can be some electrical light, right? Or it can be certain electrical light, right? Under light microscope, what all microscopes do we have? So we have four microscopes under the light microscope. First one being your bright field, right? Second one being your dark field. Then we have your phase contrast. And then we have your fluorescent. Right? And under electron microscope, we just have one, which is your electron microscope. We just have one, which is your electron microscope. Right? So we are going to talk about each one of them one by one. So let's start with the first one, which is your bright field microscope. So first thing that I want you to know is that this is what a normal microscope looks like, and it is important to know because you can get a lot of questions just asking about, they'll put an arrow on any of these structures and they'll ask you what is this. So okay, let's see. Objective and ocular lens I've already talked to you about. Other than that, there is an arm from which you hold the microscope. Then there is a coarse focus which moves the stage up and down. Then there's a fine focus which kind of fine focuses everything. So you don't have to change the whole focus all the time. There is a base. That is the base of the microscope. Then there is a light source. So that can be, as I said, either natural or electric. Usually in your light microscopes, it's going to be an electric source, right? Then you have a condenser and a diaphragm. So a diaphragm is the one that moves up and down, and that diaphragm is something that will tell you how much light is going, right? And then you have a condenser, which can also be used to see how much light you want to be focused on the specimen. Then there is a stage on which you put your slide. So that is the stage. So this is just the basic structure of the microscope and all the parts, and you need to know all these parts, right? Coming to the principle. So basic principle of the bright field is that whenever you are trying to focus something in a microscope, what is the thing that you see maximum? You see the field, right? So in your bright field, what happens that your field is bright, but the specimen is illuminated less. So your field is bright, but specimen is illuminated less, right? So your background, or you call it as, the background, the field is of the background field is bright. So background field is bright. So you will remember as background as bright, right? So that is the basic principle. Coming to the ray diagram. So this is your light source actually. So this is your light source. So what happens? You've put your light source. There is the condenser. So condenser kind of converges the light on the specimen. This is your specimen. So anything that is denser is going to reflect or refract the rays, versus anything that is liquid is going to let the rays pass, right? So there's going to be some level of deflection or the reflected rays, right? And some rays are just going to pass like that only. Okay. Then to the objective lens, what is going to happen? On the objective lens, again, all these rays are going to converge, and then it's going to converge on the ocular lens. So all these lenses are actually your convex lens. Because convex lens, if you know a little bit of physics, convex lens converges all your rays, and that is what we want in a microscope, right? So there are certain reflected rays, there are certain unreflected rays, and because of that, they are going to lead to convergence, and all the rays that are coming from the background that are illuminated, so hence your background is illuminated. So let's see this particular picture, the one on the left. So the one on the left is the unstained. So can you see the background is bright? This is bright, but this is not, the specimen is not illuminated, right? So the specimen, so this is a unstained specimen. So this is an unstained image, right? Right? So, and what are these actually? These are your fungus, which is what? Which is your budding yeast cells, right? Now, one thing that I can do to increase the contrast, remember I told you that I can do some form of staining. So that is what I have done here. I have done a staining here. So this is a stained image of the same organism. So this is a stained image. And now I have increased the contrast using what? Using some level of staining, right? So this is the same thing, but a stained way. And can you see how much better it is? The contrast has increased, so how better visible it is. So this is what you will see usually in an unstained specimen under a light microscope or a bright field microscope. And when you stain it, you will see something like this, right? Okay, coming to the next one, which is your dark field. So now, as the name is suggesting, what is going to be the principle? It's a dark field. That means your background is going to be what? Background is going to be dark. So that is why it is also known as your dark ground microscopy. So dark ground microscopy, dark field microscopy, both of them are same, because what you are doing here is that your background is dark, and what you have done is you have put some, disc here which has a central opaque area. So you have put a disc here which has a central opaque area, and you're letting the light pass through the periphery. So what is happening is that all the light is converging on the specimen, and your background is becoming dark, but your specimen is becoming illuminated, right? So that is your dark ground. That means dark background. So let's see the ray diagram here, what is happening. So this is the condenser, and in the condenser only you've added this ring which has the center portion as a central opaque area. So this is a central opaque area, right? And what is happening is that when the rays are passing, that central area is not letting the rays pass. So all the rays have to go through the periphery. So now that they're going from the periphery, what is the condenser going to do? It's going to converge everything on the specimen, on a specific denser portion on the specimen, and from that the light is going to go. So there's going to be certain unreflected light and the reflected light. So the reflected light is going to go and come where? To the objective lens, and from there it's again going to converge to the ocular lens and show the image. So here what is going to happen is that the specimen is going to get illuminated, and your unstained specimen or your background is going to be dark. So can you see this picture? Can you see how this organism is illuminated? So this organism is illuminated and your background is dark. So your background is dark and your organism is illuminated. Right? So this is an example of what? That dark ground microscopy is done most commonly in which organisms? In your spirochetes. In your spirochetes, and especially what? Your syphilis. Because they are so small, they are not visible usually. So that is why they are kind of, uh, done through the dark ground microscopy, right? So that completes our dark ground microscopy. And we're going to come to the next microscope, which is your phase contrast. So remember, I told you there are two ways by which you can increase your contrast. One is your staining, and the other thing that you can do is use a phase contrast microscope. So phase contrast microscope is used where you want to see a living organism, because staining kills them. So whenever we're going to stain any organism, it is going to kill that organism. So sometimes we want to see the live organism, we want to see the motility of that organism. And if we don't want to kill them by staining, we can use a phase contrast microscope. So what do they do here? Firstly, they use the same ring that was used in your dark ground microscopy or background microscopy, right? Your dark ground microscopy. So it is the same condenser that they were using here. Secondly, what they do is they use a phase ring. So I want you to know one concept is that whenever a specimen is put for microscopy, there are usually, different sections in a specimen. Suppose you've taken a tissue sample. So what will be there in the tissue? There is going to be a nucleus. There is going to be a cell, suppose a cell. So it is going to have a nucleus and it is going to have a cytoplasm. So most of the cytoplasm is going to be made of what? It's going to be made of water. And whatever the nuclear part is going to be there, that part is going to be made up of denser material. So anytime you throw rays at, this specimen, so there will be rays that will fall on the nucleus or the denser structures, and they will get reflected. They will get deflected. Right, and whatever passes through the cytoplasm, that means the water area, so water lets the rays pass unreflected. So they are going to pass like that only. Right, so that is the basic principle here. So what happens is that whenever you have rays, suppose this is your eyepiece. So now rays, so this is your light piece. So this is your light source, right? So what is happening here is rays are passing through the light source. Now this is your area. This is your stage, which has your specimen. So now what is happening at the specimen is that, as I said, if there are nuclear material that are denser, what is going to happen? The light rays are going to get deflected. So they are deflected or retarded, right? So they are retarded rays. So these rays are called as retarded rays. That means they are kind of deflected. So if they are going like this, they will be deflected like this a little bit. And how much are they deflected? They are deflected like this, right? And then there will be other rays that will pass through the cytoplasm, And because the cytoplasm is made up of the water, what is going to happen? It's going to go unreflected or undeflected, straight or unretarded. So they are going straight. So these rays are going straight. So these rays are going straight. Right now, what is going to happen? So now we have a phase plate. So we have a phase plate here. So what this phase plate does is that it wants both of these to kind of make a, like it wants them to collide and make a waveform like this. So it wants them to make a waveform like this, right? So what it does is that the retarded rays, so let's see like this. This is the amplitude of the light, right? So this amplitude and the two spaces between the highest amplitude is known as the lambda, right? So what is it going to do is that whatever reflected wave we had, like these ones, the retarded waves, so it was already reflected like this, it will decrease, this phase split is going to decrease the reflection by lambda by 4. So it is going to decrease the reflection by lambda by 4. Right, and the unreflected or the unretarded one, it's going to increase the reflection of lambda by 4. So when it's going to decrease the reflection and increase the reflection, they're going to form a waveform like this, and the net wave is going to be 0. So the net is going to be 0, and this is your eyepiece. So this is your ocular lens. And what are you going to see? You're going to see different kind of contrast, right? So you're going to see different kind of contrast. So it is used where whenever you want to see the live organism, that means the motility, you want to see the shape, and you want to see the inside structures clearly. So let's see here. The one photo on the left is known for a bright field microscope. So this is how you will see something. Can you see the structure, how badly illuminated it is? It is a bright field or the light field microscope, right? Coming to this is a phase contrast. Can you see the same structure? Once we had increased the contrast, we put the phase ring and then we put that ring on the condenser. And now what has happened is we can see the internal structures and we can see it so much better. The contrast is enhanced, right? So that is the use of the phase contrast. Now coming to the fluorescent microscope. So fluorescent microscope, the basic principle is that we use a fluorescent dye to tag the specimen, and then we put UV light on it, so it will make the specimen fluoresce, right? So it uses what? It uses a fluorescent dye to generate images, right? The basic principle is that when these dyes, these fluorescent dyes, are exposed to UV light, what will happen is, so they will excite, they will get excited. So these fluorescent dyes get excited, right? So these dyes, whenever I'm putting a UV light, so what is going to happen is that it is going to convert the invisible wavelength, invisible shorter wavelengths into the longer wavelengths. So it's going to convert the shorter wavelengths into the longer wavelengths, which will become visible. So it is going to convert the invisible light to the visible light, right? That is what it is going to do. So we have tagged the specimen with some fluorescent dye. Then we're going to throw the UV light on it. Because of that UV light, the dye is going to get excited and it is going to throw out the different wavelengths. So it's going to convert the invisible light to the visible light, right? So that is the whole principle of the fluorescent microscope. Now let's see what are the different parts and how does it work. So this is basically a light source from which light is being thrown, like the UV light. So it can either be a mercury lamp or a UV light. Then we have a excitation filter. So this is an excitation filter which will excite these rays. So this is a dichromatic mirror. So this is a dichromatic mirror. So this dichromatic mirror is going to deflect these rays. So it's going to deflect these UV rays on what? On the specimen that has been tagged with this fluorescent dye. So when these rays are going to come on this fluorescent dye tagged specimen, it's going to excite the dyes and the dye is going to kind of reflect the visible light. So it's going to start producing the visible light of the higher wavelength. And these higher wavelengths are then going to be crossed by the dichromatic mirror, reach the eyepiece, and then the organism will be visible fluorescenting. So like you will see the organism fluorescence, right? So this dichromatic mirror does what? It kind of reflects the shorter wavelengths, but it lets the longer wavelengths pass. So we have converted the shorter wavelength into the longer visible wavelength. So they kind of do a dual function. That means it kind of deflects the shorter wavelength and it lets the longer wavelength pass. So what are the applications? We can see all the autofluorescence. That means there are some microorganisms that have the ability to fluorescence. They already have these fluorescent dyes in them. So we can detect those organisms. Example being a parasite, which is your cyclospora. So cyclospora is a parasite which if you put under this fluorescent microscope, you will be able to see it fluoresce because it already contains these dyes. Then you can see them, you can actually coat these microbes with the fluorescent dye and see them just like we were doing it. And we can also do an antibody coated on with these immunofluorescent dyes, and you can then attack an antigen. You can make this fluorescent label antibody attached to the antigen, and then you can detect the organism, right? So these are the different applications. Coming to now the electron microscope. So the basic principle of electron microscope is that it is using electrons as a source to produce images, right? So it is using electrons, so electrons as a source of illumination, right? And also, if like we know that electrons are something that are, they will be easily deflected if there are any particles in the air. That is why we need to create a vacuum chamber so that these electrons can be accelerated. So we have to create a vacuum chamber. Right. So what does this do? So electrons are actually shorter wavelength, and anything that has a shorter wavelength has a higher resolving power. So if you have a higher resolving power, that means you will be able to see smaller structures also very, very clearly. So you need to have shorter wavelength. So electrons actually have very short wavelength. That is why the resolving power of the electron microscope is high, right? Then the advantage is that you can see the internal structures that are not visible by the light microscope. So there are mainly two types of electron microscopes. One is your TEM and one is your SEM. And what is TEM? TEM is your transmission electron microscope, and SEM is your scanning. So we will talk about both of them individually, right? So coming to the slide preparation. So what kind of slides are used when it comes to your electron microscope? They are the metal slides, right? They are the metal slides versus what slides are used in your light microscope? They are the glass slides. So we have to do some kind of sample preparation. It's not like that we can just put any tissue and just see it under the electron microscope. We have to prepare the tissue. And how do we prepare it? So we have to first take the tissue and cut it into pieces, right? We have to cut the tissue into smaller sections. Then we have to fix it. Fix it using what? We have to fix it using glutaraldehyde, right? So we have fixed it on the slide using glutaraldehyde. Now we have to dehydrate because we want to remove all the water droplets. So if there are water droplets in the sample, it's going to reflect the or deflect the electrons. That is not something that we want, right? So we will dehydrate it so that all the water is lost, right? Then we will embed it in a plastic polymer, slice that, and then you can mount it on a metal slide, right? So then you can mount it on a metal slide. So now, what is the basic principle? That electrons are fired from one end, which will fall on the specimen. So it is going to fall on the specimen, then going to fall on the objective lens, a projector lens magnet. This magnet is going to pull the electrons to itself, and then you will see it on a screen. So you will see it on a screen. So this is a screen, right? So that is your basic thing. Now that was your TEM. What is your SEM? In SEM, what is happening? It is called as the scanning electron microscope. So in this, what happens is that the electrons don't go through and through the sample. It doesn't go through and through the tissue. What happens is it goes on the surface and then it is reflected. So SEM is for your scanning. So electrons actually go on the surface and then they are reflected. So you can only see the surface structures. So you can only see the surface structures. Whereas in your transmission, the electrons are fully transmitted through the sample. So you can see the internal structures. You can see the surface, but you can also see the internal structures. Versus your SEM, the electrons are being reflected from the surface. So you can only see the surface structures, right? So that is your scanning. You're just scanning the surface. In transmission, you're seeing it transmitted and you can see the internal structures also, right? How do you increase the contrast on electron microscope? You can do some form of a staining, but you do a staining here with heavy metals. You don't do that staining with dyes. You do the staining with heavy metals. So you can either stain with heavy metals using the lead acetate, which makes these structures electron opaque. Then you can also do the staining of the background, which is what is known as negative staining. When you're not actively staining the specimen and you're staining the background, it is known as a negative staining. So here also you can use the heavy metals, and most commonly you can use the phosphotungstic acid. So you can use the phosphotungstic acid, right? Got it? So look at these structures. So the one on the left is showing what? It is only showing the surface structures. It is only showing me that there are some cilia or some things on these. It's just showing me the superficial structure. So what is this? This is actually SEM, the scanning electron microscope. And the same thing, if I see here, I can see the nucleus, I can see all these internal structures, organelles and everything. So what is this? This is actually TEM, which is your transmission electron microscope, right? So that brings us to the end of this session, and stay continued for more sessions on your general microbiology.